Hello! This video will demonstrate the decision support system for surgical treatment of Parkinson's disease. During the surgery, the data from the micro recording is saved to a file. Later, the file is parsed by this application. Finally, it performs the decision support classification. First, we select the directory where the data has been saved from the medical system. We select whether we would like to parse data from the left and right hemispheres, from the left hemisphere only, or the right hemisphere only. Here, we will only parse the data from the left hemisphere. After picking the directory and selecting the left hemisphere, we click Parse. Now we can see the data that is passed from the depth by each electrode. The left anterior electrode is processed depth by depth. We can see those depths. Here, we see grayed areas that represent the forbidden amplitudes for the spikes. Here, we can see the average shape of the spikes, and on the right, this average shape is presented as a spline. We can see the spikes detected in the recording are aligned to the maximal amplitude value. Each file represents a recording from a given depth. It is parsed, and when all the recordings from a given electrode are processed, the data is sent to the database. Now, the first two electrodes have already been processed, and now the third and last electrode is being sent to the database. The whole procedure of parsing is quite a fast one. It only took slightly above 1 minute to complete. Now we can move to computing the statistics. We select the patient, and we can see that for this patient there is only data for the left database, and we select the computation of spike statistics. This computation takes a very short time, as during the parsing procedure, the spikes have all been detected and sorted. As you have seen during the detection, there are different shapes of the spikes. When the electrode is inside the brain, often it is in the vicinity of several active neural cells. They produce the action potential, the spikes, that you have seen in the previous stuff. Each cell produces a slightly different shape, so when we look at the shape, we can discriminate using it between the spikes from each cell. The spikes from each cell can be thus treated separately. When we complete the spike statistics, we can go on to computation of the background statistics. The background statistics take a bit longer to complete, as the whole dataset has to be processed, not only the spike occurrences. During the calculation of the background attributes, any artifacts that are present in the recording have to be removed. The artifacts are high amplitude samples within the recording, and they are produced by the patient speaking during the surgery. The patient is conscious, so it is quite possible. By someone else speaking, the patient is hearing him, and this hearing of him is represented within the recording. Or by someone touching the stereotactic frame attached to the patient's head. So it is quite possible that there are artifacts. As you can see, they can be quite numerous. These artifacts have to be removed prior to computing the background attributes. Without this removal, those attributes would be incorrect. When we have finished calculating both the spike and background attributes, we can pass to the classification. Classification can be done in two ways, in an automatic way or in a manual way. We will choose the automatic classification. Automatic classification is done using the Weka software in the background. We can click this button, and in this moment, the data from all attributes, both the spike and background attributes, are passed to Weka. Using the trained classifier, the recordings are classified as to whether the recording is coming from the STN or not. It has been completed, and now we can also go to the manual classification. This is an optional step, but we can adjust the classification result or make our own completely. After clicking this, we can see that for this patient's anterior electrode, we have data from several depths. Each depth is represented by a row in this array, this table. Depths that have been classified as belonging to the STN are shown in the green color. You can adjust the selection by adding some selections and subtracting others. After any adjustment, you can save this result, or you can classify again using Weka. This is a way to return to the original state. In any given moment, we can render the output of those attributes by clicking this button, and we can see the results of the rendering. The attributes are shown on the bottom of this window, and by clicking each of the small pictures, the user can see it in a larger way. As we see at this moment. But as to discussing the meaning of those pictures, we are going back to the Results tab. Here, we can choose the patient and the left hemisphere. Now we can see the attributes that have been calculated, and we can see how they changed over the depths as the electrodes passed through the brain. The first attribute is the attribute that is called spike meta power. This is the power of the signal that has been artificially constructed from the spike occurrences within the original signal. In this way, any background data is discarded and only the spikes are accounted for. The anterior electrode is shown in red, the central electrode is shown in green, and the medial electrode is shown in blue. There are 4 spike base attributes. 2 attributes show the spike occurrences per second. Essentially, this means how many spikes per second on average are found within this recording. This can be shown here for all cells near the electrode or for the best cell near the electrode. As we have stated, it is possible to discriminate whether a given spike belongs to this cell or another cell. In this way, we can treat those cells separately and select the best one. This is for the best cell. Moving on to the best ratio. The best ratio is based on the distance between spike occurrences. The distances between spike occurrences can be shorter or longer. Some of them are shorter than 33 milliseconds, some of them are larger. This attribute is the percentage of all interspike intervals that are shorter than 33 milliseconds. This attribute is on a scale between 0 and 1. Finally, here is the best ratio for the best cell near the electrode recording tip. The last groups of attributes are the background attributes. The first one is the 80th percentile of the amplitude modules. You can see immediately that background-based attributes give a much clearer picture of the results. You can see the area where the structure we are looking for, the subthalamic nucleus, is located. We can see from this picture that this area of high-value attributes is the area where we would expect the STN to be. It is also in agreement with the fact that neurosurgeons calibrate the device in such a way that they expect to find the nucleus at depth 0. In this case, they hit the target perfectly. So, at any given moment, we can also check the box marked classified, and at that moment, parts of the lines of the charts will be shown with a bold line. After clicking, we can see that for the anterior electrode, the STN is predicted to be in this area. For the medial electrode, we expect the STN to be from there to here. In a similar way, we can look for the other background attributes, our root mean square, low frequency background, and high frequency background. Low frequency background is the power of the signal in the frequency band between 0 and 500 Hz. And high frequency background is for the band between 500 Hz and 3 kHz. The last group is based on this group, and it is simply a moving average with width 5 and with padding 1. The padding 1 is selected because the background attributes are normalized. They are normalized in such a way that for each electrode, the average from the first 5 depths is calculated, and all values of the selected attribute for this electrode are divided by those averages. So, the level of 1 is a typical level of low activity outside of the STN. So, a padding of 1 is safe here, because padding should reflect the areas that are outside of the STN. We can also select the patient, select the hemisphere, the electrode, for example, the anterior electrode, select a depth for this electrode, for example, -1000, that is -1 millimeter, and play the sound that has been produced from the recording at this depth by this electrode. Those strange sounds that you might have heard at the beginning of the recording, those are artifacts. Those artifacts have been produced by someone touching the stereotactic frame attached to the head of the patient during the operation. Even the slightest touch to the stereotactic frame produces very loud artifacts. Finally, after calculating all of the attributes, we can go to the report. After selecting the patient, we can click Report, and after a moment, we can see the report shown in Microsoft Word or in any other program capable of showing a rich text format file. The main point of this report is this table. The table shows a row for each electrode, and for each electrode we have columns representing depths. The depths at which an electrode passed through the STN are shown as a red S, and the other depths are shown as a blue M. S stands for STN, and M stands for miss. We can see now that the best electrode is the anterior one, because it passed through the STN at depths from -2000 to 2000. The medial electrode passed through the STN from -2000 to 1000, which is slightly shorter, but we are looking for the electrode which has the longest passage through the STN. In this way, the table shows the neurosurgeon which electrode is the best and which electrode is the second best. Of course, the electrode selected as the best one by the program is not always used for the implantation of the stimulation electrode. This is because in some cases the stimulation of the best spot causes some adverse side effects. In this case, it cannot be used and the other electrode has to be used. In this way, if the anterior electrode causes some side effects, the second best one to try would probably be the medial one. This is the table that is shown to the neurosurgeon during the surgery. Below this table, we can see the chart showing the described attributes at the given depths for electrodes. So we can see that the RMS for the medial electrode at depths from minus 10,000 to 5,000 is shown by the blue line, the central electrode is represented by the green line, and the red for the other electrode, and so on. For the 80th percentile, it's this chart, and for the low frequency background and high frequency background, the proper charts are shown. Below, you can see the charts for the moving average of background attributes. Finally, the charts at the end are showing the spike-based attributes. You can see that the attributes that are background-based are much clearer and give much more evidence as to where to expect the STN to be than the spike-based attributes. This is because spike-based attributes rely solely on the detection of the spikes. In some cases, within the STN, where normally one would expect high spiking activity, the electrode happens to be in an area without any active cells near it. So, we can obtain a false negative result. We can think that we are outside of the STN when in fact we are inside of it. It is also possible to have it the other way around. There are certain areas outside of the STN that can produce a very high level of spiking activity. For example, at the negative 10,000 level, it is possible to obtain a very high level of spiking activity. This spike activity, of course, does not mean that there is part of the STN at this spot. No. It is that there are other brain structures that are also highly active. In this way, it may also cause false positive results. This is why the background attributes are much, much better than the spike-based ones. Thank you. This system was developed by Dr. Konrad Ciecierski in his PhD thesis written under the direction of Dr. Zbyszek Ras, voiceover provided by Laurel Powell. Thank you.